Hello everyone, I'm Farmina. Today I'm going to present on the topic um, Discovery and Revision of Arabidopsis Genes by Proteogenomics. So my teammates are Sultan Talha and Sonam. To start with, Arabidopsis gene is a model organism used to determine as a defense mechanism for plant pathogen resistance. It is more often used in genetic experiments for its little non-coding DNA region and also short generation time, and it is best annotated plant genome. Some drawbacks which includes errors in the reading frame, protein per day. Going on to the next slide, detection of gene model errors in the experiment. For this, Arabidopsis proteins are isolated from different tissues, and then tandem mass spectrometry is done to detect tryptic peptides. Then 1,44,079 peptide sequences were determined, of which 40% annotated genes were confirmed, and the results which showed was 18,024 novel peptides were identified. That suggests 13% proteome was incorrect or missing, and amongst them 1473 gene proteins were corrected, leaving excluding 1 to 4% proteins from identification. Now this slide, here I'm going to discuss about the modules of mass spectrometry and how the process goes on. Here the sample which is converted into ion source, and then from the ion source it's delivered to the mass analyzer where the ions are sorted out by using electromagnetic fields. In the detector, indicator quantity is determined, and for the data analysis, data are collected for the data analysis. Now I'm going to talk about the workflow of the system. Like first of all, the databases are collected and input into the computer system for inspection. From here, 1,44,079 peptides were determined, of which some of them are having the novel peptide. If it has the novel peptide, the novel hits are determined and the clustering peptides are also determined. If there is no novel peptide, then the genes modules are refined using tier 7 hits, and the genes and the peptides are classified. For example, the peptides, proteins, and the splice peptides. For determining the gene location, it's also classified according to the novel exon skipping, boundary change, frame correction. Here's the results given in the flowchart. Now I'll pass to my next teammate to discuss the rest of the topic. Thank you.
Thank you, Farmina. So now I'm going to talk about some results of the experiment. So discovery of novel gene. The identified cluster of 13 located peptides does not overlap current gene model where the prediction track displays the single exon gene model generated by Augustus. So what is Augustus? Augustus is a tool for finding protein coding genes and their exon intron structure in genomic sequences. It does not necessarily require additional experimental input, as it can be applied in so-called ab initio mode. However, extrinsic evidence from various sources such as transcriptome sequencing and annotations of closely related genomes can be integrated in order to complete the accuracy and completeness of the annotation. Augustus can be applied to single genomes or simultaneously to several aligned genomes. From the sequence that is predicted, it is found that strong homology is established between thylakoid lumen family protein and also similarities to protein that are in grapevine and rice. So predicted transposable element being overlapped by peptides. We also noticed that several interesting structural features of the intergenic clusters. First, a significant fraction, almost 64% of intergenic clusters overlap annotated pseudogenes or transposons. For an example, as a translated pseudogene is at a locus and the name is ATRLP18, which is a receptor-like protein which has high homology to disease resistance proteins both in Arabidopsis and other plants. We identify 5 such peptides, 3 of which are uniquely located at the same locus, confirming translation. We know that pseudogenes do not produce proteins, but transposons can contain active protein coding genes, but they are not typically included in the proteome. We find evidence in transposons of translated proteins that are unrelated to transposon activity. For example, we identified three peptides at this locus, naming 84G07947. In the picture, we can see that. Although annotated as a pseudogene in tier 7, it has been reclassified as a transposable element gene at tier 8. The genomic region containing these peptides has high similarity to the ubiquitin-like protease family in Arabidopsis, suggesting this may be a gene traveling as cargo with the transposable element. Refinement of gene. So in addition to novel genes, they also discovered peptide clusters overlapping annotated gene models, suggesting refinement of existing annotation. For example, a new exon, exon boundary change, exon skipping, or modified translation boundaries. The refinement events can be classified according to their type, location, and the transcript being modified. So a majority of the events here are novel exons, like of which 314 are located within introns and 207 are in the untranslated region of the tier 7 gene model. It is difficult to determine whether a new transcript predicted by Augustus is a refinement of a gene model or an additional isoform of an alternatively spliced gene. To better distinguish between these two cases, they have compared each new transcript and the tier 7 transcripts to all available cDNA evidence. So they have identified 70 cases in which the annotated reading frame is different from the observed peptides. Assignment of reading frame is particularly difficult for nucleotide-based genome annotation. However, proteomic evidence unambiguously defines the frame of translation. The 70 frame corrections are supported by multiple peptides and extensive homology to other proteins. So for example, here the 81G63500, a protein kinase, which has four novel peptides as in the annotated 5' untranslated region. These peptides point to a large expansion of a gene and a misprediction of the current first exon. Four novel peptides map within the 5' untranslated region and the first exon are shown in the picture A, and the B picture shows that zoom of the region shows that the current first exon is out of frame with the peptides. And in C, we can see the sequence alignment with the Arabidopsis and grapevine proteins supporting translation in the frame supported by peptides. So to estimate the strength of the annotation, spectral counts are also a strong indicator. Like 219, 291 peptides have higher spectral counts than 50% of all peptides identified in this study. The intergenic peptides indicate novel coding regions that may have produced a single detectable peptide for several reasons, including protein composition and protein length.
Hi everyone, I'm Sonam and today I'm going to talk about challenges and importance of proteogenomics. Recent advances in mass resolution and the availability of software tools to analyze spectra make mass spectrometry the tool of choice for proteomics. Nevertheless, technological limitations create many challenges for the approach. The first challenge is that the sampled peptides are biased toward the more abundant proteins in the cell. Second, incomplete fragmentation patterns and spectral noise smudge the fingerprint and introduce errors in peptide identification. Third, the post-translational modifications change the mass and pattern of the fragments, making identification harder. By investing in proteogenomics to complement more traditional cDNA and EST data at the onset of genome annotation, a more complete and accurate proteome can be achieved even in the early releases. They provide proteomic evidence for 778 new genes and refine 695 current gene models using the reference annotation from TAIR7. Recently, TAIR has released the next version of the genome proteome, TAIR 8. Only a small number of their novel peptides, 3%, appear in the TAIR 8 release, indicating that the proteogenomics approach is complementary to computer-based annotation.